Good morning. My name is Shreshth. If you're wondering what that strange constellation of consonants mean in my name, I pronounce it Shreshth, and I have the privilege of leading the machine learning group at Deep Genomics. We're a company based out of Toronto. We also have a footprint in Boston. We recently opened an office near Kendall Square last year. Today I'll be talking about end-to-end genetic drug development, which enabled by what we call the AI Workbench. And I'll be illustrating some examples of how we've used machine learning to enable this use case, and we've tried to use machine learning throughout the pipeline of drug development. So the first thing to notice here is like why are we using machine learning in this context? And there's really three ingredients, and Deep Genomics is really sitting at the intersection of these three things. The first being, for the first time in history, we have, through RNA technologies, an opportunity to have a programmable medicine. And this has become clear over the last few years about how transformational RNA therapies can be, and really this fits very well into this paradigm of being an information drug. The second thing, and which struck me the most when I joined this field 5 years ago, was how much data exists for RNA biology. It's really one of the few fields where it's really amenable to the kinds of techniques in machine learning and deep learning specifically, which are well suited to take advantage of all of this data. And really the enormity of the data that we have for RNA biology makes artificial intelligence one of the most suited ways to actually entangle or disentangle all of this data and use it to generate new insights. And our mission is to program the best RNA therapies for any gene in any genetic condition. And over the course of my talk, I'll try to illustrate how we try to achieve this with some specific machine learning methods. So the focus of our company is to use AI for both genetic target discovery using genomic data sets, and also for predicting the effects of, actually to find the therapies for those diseases as well. The modality of drug we use is called a steric blocking oligonucleotide, which fits very well into this information drug paradigm. And the advantage of our approach is really what we call the AI Workbench, where we expand the space of potential target genes and targets using AI to uncover the sort of dark space of potential therapies, and also consolidate all of the learnings into a platform. Our disease focus, when we started, it was for rare dominant haploinsufficiencies. And as we're getting more sophisticated with our target discovery methods aided by machine learning, we're now also moving towards more complex disorders. Our focus has been to sort of take advantage of both the AI ecosystem in Toronto and the clinical ecosystem in Boston. And we have a footprint in both of those places. Our portfolio is early but rapidly advancing. We have targets in the CNS as well as metabolic disorders, and we have a few programs in partnership with BioMarin. The next question is, of course, why did we choose RNA biology, and what is really different about the approach at Deep Genomics? So the first fact that I would like to highlight is that, like, if we approach the problem of screening compounds for just a single gene, if you know for example that you want to increase the expression of a given gene, and the only task you want to solve is to actually screen compounds to find the ones that have the desired effect. Without the use of AI, without sort of highlighting where you should place these compounds, you would need an enormous number of compounds to screen just to find top 3 out of the 10 hits that we identified using our platform. And to sort of illustrate this further, on the right here, I'm showing the the number of compounds that you would need to screen to find 90% of hits for a given gene. This is based on a few representative genes from our screening programs. And the use of the AI Workbench allows for a 15-fold improvement in screening productivity. And on the left side, we're showing that by encapsulating all of these mechanisms through machine learning models, we're able to expand the target space for genes that are amenable to expression increase, and which is 2 times higher than what you would be able to achieve with a traditional approach that other companies are focused on. So the reasons why we've chosen steric blocking oligonucleotides as our modality are sort of fourfold. One is that you can really look at multiple ways of achieving the same effect. So ultimately, our drugs are just like a sequence of letters. And by changing that sequence, you can achieve many different effects. So you can increase the expression of a gene. You can decrease the expression. In some cases, by altering splicing, you can change the actual protein that's produced. So by targeting the different regulatory pathways, you can achieve all of these different effects, which makes it a very diverse modality. It's also very specific. You can have very targeted on-target effects. And it also has like good properties. It's safe and tolerable. And there's been a lot of great work done by the community over the past decades to develop oligonucleotides into a safe modality. And here I'm really highlighting the sort of the difference in our approach and like how we view the platform way of thinking for drug development. In a sort of traditional way of thinking about it, you would have this sort of waterfall approach of going through the different steps of drug development, and each of them are somewhat disentangled in that there aren't that many common elements, and the risk is sort of carried forward. With the platform way of thinking, which is on the right, we have this central element, which is our AI Workbench, which is enabling all of these steps and is also evolving over time with the data that we generate in each of these steps. And by doing this, we can de-risk a lot of these things beforehand, because you can predict into the future. You can look into ways in which your therapies could potentially fail at later stages. And by predicting exact mechanisms, you're able to do this very targeted de-risking. For instance, if you predict that a variant is a mutation which is a missense mutation is predicted to alter splicing, you can then test that in an assay. And if you also want to predict then what effect you would observe by placing by testing that mutation in mice, you can develop a mouse predictor for splicing, and then you can look into the future of what you would plan to see in sort of later stages. So this is just another reminder of the way we're sort of thinking of our drugs as digital information. The sequence of letters that you see here is actually one of the drugs that was predicted by our platform. And just by the striking thing to think about is like just by changing this sequence of letters, you can have drastic effects. You can upregulate or downregulate a gene. You can pick which gene you want to target, and you can change the actual protein that's produced. And really the 3 key properties that make this approach very suitable for artificial intelligence is the digital information aspect, which I mentioned. There's also massive data sets for all of these different modalities, which make it very suitable for AI. And the third key principle is by modeling the sort of very fundamental processes of RNA regulation, you can learn generalizable and causal rules. So if you train a predictor for a very low-level cellular mechanism, for instance, like RNA splicing, you can then expect it to generalize because you know exactly the mechanism of your drug and the mutation that drives it. So now I'll give you a bit more detail on what actually is under the hood, what we call the AI Workbench. Before I do that, just a sort of reminder for most people about the central dogma and where this sort of AI fits in here. In this subsystem here, you have your genomic DNA, which is then transcribed into pre-mRNA. It's spliced into mRNA and translated into proteins. There's many steps that happen throughout this process. And this is a subsystem that's very well understood and very well conserved across species. And all of these processes that are highlighted with the little green logo, all of these correspond to a machine learning predictor that we've built for that process. So anytime you have a disruption in that process, you can, by, let's say, a genetic mutation, you can pass it through the predictor, and it gives you a sort of readout of how that process itself is disrupted. And then you can target your therapy to actually reverse the effect of that process. So all of the steps here, starting all the way from transcription factor binding, going into sort of post-transcriptional regulation, correspond to one of the predictors that we've built. And these are just some of an illustration of the types of data sets that exist for all of these processes. If you put everything together, there's petabytes of data that's available for RNA biology. Not only are we talking about data for therapeutic compounds, which are highlighted in orange here, which is oligo data, but there's also sequencing data, what we call RNA biology data. There's evaluation data sets for variant data, like massively parallel reporter assays, which we can use to test our models and fine-tune them. But put all together, for each of the processes that I highlighted in the previous slide, there's an abundance of data that's available. So a key insight is if you connect the previous picture of regulatory mechanisms, you can, if you build accurate predictors to model them, you can then also predict inefficiencies in all of those processes. For instance, if you have a model that predicts RNA splicing, you can then also predict when splicing is occurring inefficiently. For instance, if an exon is not being spliced correctly all of the time, there's a small population of cells which the exon is not included, which is leading to lower expression. You can then intervene into that process with an oligonucleotide and increase the efficiency of that process, and thus increase expression. So with these models, we have a unique opportunity to intervene in all of these processes and sort of uncover inefficiencies when they exist. And another key aspect of connecting all of this together is automation. If you look at a new gene target, there's hundreds of thousands of potential compounds that you could screen, and each gene is sort of very heterogeneous. It's spliced in different ways. There's different polyadenylation isoforms. There's differences in the transcription initiation sites. And if you don't build sort of automation approaches which automatically highlight all of those potential opportunities for upregulation, each of the genes becomes a sort of new unique target, a new unique challenge to solve. And what really enables our productivity and data integration is we automate the process of highlighting these target areas and selecting these compounds, which makes it possible to, in silico, then test hundreds of thousands of potential compounds and end up with only, let's say, a list of 1,000 compounds that need to be tested in the lab, which you can do in a matter of a few months. And our AI is trained, just to give you some examples, it's trained to identify both disease-causing variants and also therapeutic oligonucleotides. And like you can use basically the same predictor, because what we're predicting is really molecular phenotypes. It's a low-level information of RNA regulation. It's not really uniquely tailored or sort of designed specifically for either of these applications. You can fine-tune the predictor to achieve both effects. For instance, again, going back to the splicing example, you can see which changes in the sequence causes a decrease in inclusion of an exon, and then also use the same predictor to then in silico tile which SBOs would then restore the inclusion of that exon. And you can do this across different RNA mechanisms, and this approach of using the same predictors for both predicting variant effect and oligonucleotide effect is something which is central to our approach. And to give you a sort of concrete example for a therapy which many of you would be familiar with, for spinal muscular atrophy, it took around 15 years to come up with Spinraza, which was a transformational drug. And if you put it in the context of our approach, we, over, let's say a few hours, we could run our splicing predictor. And we actually did this of like running all candidate exons. And the target for Spinraza came up in the top 10% of that list. And in the sort of bottom slide here, on the x-axis, I'm showing different locations across the SMN2 exon 7. And on the y-axis is the predicted effect of a potential therapy. The higher you go, the more efficacious the compound is predicted to be. And we've pointed out Spinraza as one of the peaks. And all of this can be done in an automated fashion, end-to-end, and which sort of enables not only reliability, but also you can repeat this process and then use this to find more drugs like this. Just give me a second. Can we go to the next slide, please? And to give you another example of a recent predictor that we developed, we've used techniques which are sort of now very common or ubiquitous in the language and vision community, namely using transformers to build these sort of massive models which take in over 200 kilobases of genomic information and predict thousands of tracks at the same time. So in this example, we're taking literally the raw DNA sequence and predicting hundreds of these RNA mechanisms simultaneously using the same predictor. And what this allows you to do is, because you're forcing the model to learn a causal signal, because you're literally only providing it the DNA sequence, you can then look at perturbations in the sequence and see what effects it'll have on those RNA mechanisms. So over the next slide, we've looked at pathogenic variants in ClinVar, which is a database which is consolidated from clinicians, and we looked at our ability to identify pathogenic mutations in the untranslated region, both for the 5' and 3' UTR. And because we're sort of looking at all of these RNA mechanisms together, our models are able to learn a sort of general code of how sequence maps to gene expression, and sort of combine all of this information to give you a single score of perturbation. So if we just use the model to score these mutations that have been reported, we are able to distinguish the pathogenic ones from the benign ones with sort of great accuracy. Here we're comparing our predictor to Informer, which inspired our sort of idea for this model, which was work done by DeepMind. But crucially, because we sort of targeted this model to focus on RNA biology, we are able to sort of capture these RNA regulation mechanisms more accurately than Informer, which was sort of focused more on things like DNA accessibility and cage. To give you another example of how this fits into the oligo design paradigm, we've designed a predictor for microRNA binding, where we've taken microRNA binding information from over 26 different cell types across mouse and human tissues. And we've trained a predictor to identify sites where you have potential microRNA binding. So on the right-hand side, the two plots that I'm showing are microRNA targeting oligos that were reported in 2 papers over the past decade. And on the top plot is the hotspot, so target regions predicted by our microRNA predictor. And what I want you to notice is that the top region that's predicted really very well coincides with the SBO that was reported in the paper. So if you were to go and sort of do this experiment, if someone gave you this gene and told you to find some locations to place an SBO, the first thing you would do is like look at this predictor, find the sort of top regions that are annotated, and you would place an SBO and you would be able to find it. And we've shown this, I'm showing 2 examples here of 2 similar studies which did the same thing. The orange plot is using target scan, which is a sort of non-deep learning based, a sort of rule-based approach for predicting microRNA binding that's commonly used in the literature for finding microRNA binding sites. Using these sort of deep learning predictors, because you're consolidating a large amount of information and you're trying to make it very precise, you can make these sort of very pinpoint predictions and give you like very single base pair accuracy at predicting these sites, which is not really available if you use the sort of low information predictors that are not using as much data. So yeah, I'd like to sort of talk a bit about what's in the future. I gave you some examples of predicting expression increase. We've also done a lot of work on splicing. It's one of our sort of more mature prediction technologies. There's a lot of interesting work going on in the company right now for predicting oligochemistry and how we can use machine learning methods which are sort of uniquely suited to actually model the oligonucleotides not just as a sequence, but as an actual graph, incorporating its sort of backbone structure as well. And I'm looking forward to the work that we'll do in the future to actually model the oligochemistry as well. On the genetic side, we've focused a lot on sort of initially on rare Mendelian disorders, moving on to sort of dominant haploinsufficiencies. But in the future, we're also sort of incorporating data that's being generated from biobanks and trying to use those as sort of potential sources of finding complex targets or targets for complex diseases, like Alzheimer's, for instance. Another key piece of work is also trying to de-risk the sort of later stages of drug development, like the in vivo translation. Many of the approaches that I mentioned, they're sort of agnostic to where the data comes from. So you can imagine building predictors which predict both the mechanism in humans, but also mice and in non-human primates, which lets us sort of predict if a target region that we identified in an in vitro setting, one, will it translate to primary cells? Will it then translate to an in vivo setting in small animals? Will it also have the same effect in non-human primates? And crucially, will it retain the same effect in humans? And regardless of any type of drug modality, there's always a dearth of actual human data that's available. And I think that these approaches of using machine learning to actually consolidate all of these mechanisms into shared neural network representations is a very unique way of actually de-risking the final thing that we all care about, which is actually improving lives, improving the health outcomes for humans. So on the right here, I'm just showing you some cool pictures from our high throughput imager. So these are images of mouse neurons. And we've really started just doing this work of actually testing our compounds in mice and using these high throughput imaging technologies to see what actually our compounds are doing. And I'm really excited by this approach, especially because it sort of really brings home the central idea of what we're trying to do is actually predict things in vivo. And just the key takeaways that I would like to highlight are sort of the intersection of the three things that make Deep Genomics unique is use of RNA therapies, use of large amounts of data, and artificial intelligence as the central flywheel which is driving this innovation. And yeah, with that, I would just like to close my talk and remind you of, or sort of just mention our mission, which is to decode genome biology to program life-changing medicines. And I will just like to share my hope for the next 10 years is I think AI will play a transformational role in how we view medicine, how we view innovation in medicine, and hopefully we'll be able to intervene at earlier and earlier time points in disease progression. Thank you.